Thank you, Brett. Thank you, organizers, friends and colleagues, and everyone else. I'm pleased to participate in this symposium. It's already been quite interesting with lots of new ideas. The title of my talk, as assigned, was Missing Microbes, and so I will use that title. And just tell you just a little word about the book that I wrote called Missing Microbes, which this was my baby. I started thinking about this particular book around 2005. It was written years later, and I didn't have a chance. Edited and then published in 2014. So it's been 4 years that it's been out. You can see that it is out in French. It's been well translated and has gained the popular attention, and that was my hope. The hope was not just to speak to scientists and doctors, but to speak to the general public about an issue that concerns us all. So let me continue with the theme that our first speaker began with, which is the change in our macro environment. And this is a view of the ice sheet in Greenland, 10 years apart from space. You can see there's been a big reduction in the amount of ice. We take this as one of the many pieces of evidence about global warming. And we could define global warming as a change in our macro environment as a result of human activities. And what I want to talk about is a change in our microenvironment that I would argue is of comparable scale and probably happening even faster. So this is really about ecology and about the ecology over the lifespan. And I want to cover these 5 topics: the scale of the problem, a theory about the causes, the practice of prescribing antibiotics, evidence that antibiotics play a causal role, and then finish with solutions. So what I want to address is the fact that since World War II, a number of diseases have risen dramatically and rather abruptly. These might include juvenile diabetes, which is doubling every 20 or 25 years, diseases of the esophagus, like reflux esophagitis and adenocarcinoma of the esophagus, which follows this, asthma, as we know. There are about 10 different major diseases, and the question is, what's causing them? Is there 10 diseases, 10 entirely different causes? Or perhaps there is one thing that is underlying all of them. And my hypothesis is that what is underlying them is a change in the human microbiome. And I hope to show you evidence for that. So I first want to begin by focusing on one of these epidemic diseases, which is obesity. This slide are 3 graphs from the Centers for Disease Control looking at obesity trends in U.S. adults. Here in 1989, there's no state in the United States where more than 14% of adults are obese. In 2010, there's no state with less than 20% obesity, and the national average is about 30%. What's remarkable is that it is happening all over the United States, and that the distance between the first map and the last map is only 21 years. Something very powerful is changing human physiology. We know that obesity in adults begins in childhood. And this slide are data from the N. Haynes population-based studies in the United States over about a 35-year period, looking at trends in U.S. children and adolescents. And you can see the way the trends are going, even in the youngest children under the age of 5. This is, again, representative of a wide body of data. Now, that's the United States. But in fact, obesity is rising all over the world. Here's a recent review which looked at the percentage of children under the age of 5 who were overweight and obese. Here it is in developed countries, reaching about 14% of the children in 2015. In developing countries, the rates are lower, which is not surprising, but you can see that the rates are going up as well. And if you think about where the rates are now and where they're going, it's actually very similar to what was happening in the developed countries, but it is lagging by about 30 years. It's the same curve. Now, this is based on the rates, but this slide is based on the numbers of children under 5 who are overweight. And here, it looks a little different because in 2015, 80% of the children in the world who are overweight are in developing countries. And this number is going up rapidly. In a few years, it will be 90% of the children. So this is not an epidemic, this is a pandemic. Obesity is spreading all over the world, first in the West and then in the developing countries. It's not the only disease that is showing this kind of bimodal activity. Here's a graph looking at worldwide trends in inflammatory bowel disease, first recognized in the 19th century in the developed world and increasing progressively, and then beginning maybe a century later or beginning to be recognized in the newly industrialized countries. The same kind of time lag. And this, again, we might think of this as related to when modernizations occurred. So what could be causing this? Well, I mentioned the microbiome. I want to introduce 3 concepts that underlie what I'm discussing. The first is evolution. And I'd like to show this slide from Howard Achman and colleagues who looked at the evolutionary relationships of wild hominids. And in their study, they found, looking at host genotypes, they found a phylogeny that had a particular topology. And when they looked at the phylogeny of the fecal microbiota and the topology related to that, they found the same kind of branching pattern in the species, in the microbes that were seen in the host. And this kind of congruence is evidence for the vertical transmission of the microbiota from mother to child, from generation after generation. And it is also consistent with a concept of coevolution of microbes and host. And what's important here is that the time frame that is encompassed in these graphs is about 8 million years. That's how long we have had this congruent evolution. And we think it's much longer, but that's what's captured in this study. Second is a concept of equilibrium and interaction. This is from work I did with Denise Kirshner, a mathematician. Our idea of co-evolved colonizing microbes and their host are ones that are interacting, in which the microbe is providing chemical and physical signals. They are talking to the host is conversing back via the same parallel kinds of signals. We've modeled this as a dynamic equilibrium. Denise has shown that it is robust and resilient. And what I want to talk about today is what happens to the physiology of the host when the co-evolved microbe is lost, when it becomes extinct. The third concept is a concept of age window. And there, the question is, when does the adult gut microbiome become established? And I turned to work by Tanya Yatsenenko and colleagues at Jeff Gordon's lab involving Maria Gloria Dominguez, Rob Knight, and others. It was a comparison of healthy people in the US, Amerindians in South America, Malawians in Africa. On this graph, each circle is the composite of the gut microbiome in one individual. The x-axis is age and childhood. The y-axis is unifrac distance between children and adults. This is a metric of dissimilarity. And what the data show very clearly is that when babies are born, the population structure of their microbiota is very unadult-like, but that gradually and progressively it becomes more and more adult-like until it is. And what was remarkable about this study is that the age of transition is the age of 3. So the first 3 years of life is when the microbiome is the most dynamic, when it is developing its adult structure. It is also the period of time when babies are developing their metabolism, their immunity, and their cognition. So let's go back to this issue of transfer of the microbes from mothers to children. We humans are mammals. So we were born in a womb that is sterile or mostly sterile. Our first big contact with the microbiota occurs when the water breaks, when the baby descends through the birth canal and is covered by the mom's microbes. Baby is swallowing the mom's microbes. Now there's skin-to-skin contact between mom and baby. The baby's mouth full of microbes inoculates the breast, and now the microbes in the milk become the substrate for the foundation of the GI tract microbiota. Moms kiss babies, they lick babies, they pre-masticate food. Lots of redundant mechanisms for the transfer of microbes from one generation to the next. That's how it's always been for mammals. But in modern times, moms are different than they used to be. Moms are living in a world of antiseptics. Moms have taken antibiotics, including during pregnancy. Moms are subject to a variety of antibacterials in their food, in their water, on their skin. And of course, babies aren't the same as they used to be, because babies may be born by cesarean section. They may miss the travel through the birth canal. In the United States, 1 baby out of 3 is born by C-section. In some countries in Europe, it's close to 1 baby in 2. Babies are bathed extensively. They are fed formula, which only superficially resembles human breast milk without the advantages of millions of years of coevolution. And of course, babies are getting lots of antibiotics. So on the basis of these ideas, over the last really 20 years, I've been developing an idea which I now call the theory of the disappearing microbiota. And in essence, it has 2 concepts. The first is that changed human ecology has altered the transmission and maintenance of ancestral microbes, and that affects the composition of the microbiota. And second, that especially important are microbes that are usually acquired early in life, since they affect a developmentally critical stage, that stage in the first 3 years of life. Now, about a decade ago, the late Stan Falco and I enlarged this concept, which is shown here, the effect of maternal status on the resident microbiota of the next generation. Our idea is that ancient moms had an ancient microbiota, which they would pass on to the next generation. But if they lost microbes and couldn't regain them horizontally, then the next generation would be born at a deficit, and so on and so forth. And in fact, this is our conception for what has been happening over the course of the 20th century into the 21st century, that instead of the microbiome resetting with each generation, that the loss has been cumulative, resulting in a progressive disappearance or extinction of organisms. So at this point, this is theoretical. We don't know the answer to this, but there is already evidence developing that supports this hypothesis. In the first case, I'll turn to the story of Helicobacter pylori, which until recently was the dominant organism in the human stomach. And this study by Urita and colleagues, looking at 3 generations in Japan, shows the same kind of step that we had anticipated. Other work has looked at the overall diversity of the microbiota. And this is the same slide that Maria Gloria Dominguez showed from her work, comparing kids in the US, transculturated people, and an uncontacted group of Amerindians, showing that the highest diversity is in this group, the lowest is in the Western kids. And in fact, based on this metric, it is consistent with the hypothesis that people in the United States may have lost as much as 50% of their diversity. So the question is, what's the significance of this, and what might be causing it? And I want to turn to one factor. I think there are many factors, but I want to turn to one factor which I think is particularly important, and that is antibiotics. All of us here recognize that the discovery of antibiotics was one of the greatest discoveries of the 20th century. Humans have been taking antibiotics in pharmacological doses since the late 1940s, so we're now about 70 years into the antibiotic era. Antibiotics have saved innumerable lives, Antibiotics have revolutionized every phase of medicine. And as a consequence, health providers have been using antibiotics more and more and more. How much more? Well, as Henrik showed earlier, a recent estimate is that there were 73 billion antibiotic doses taken by humans annually. That's 10 pills of antibiotics for every man, woman, and child on Earth, and those numbers are going up. In the US, the CDC has actually counted the number of courses. In 2011, it was 262 million courses of antibiotics. That's a rate of 842 courses per 1,000 population. That's 5 courses for every 6 people year after year. In children, based on the CDC data, we could estimate that the average child by the age of 2 has received nearly 3 courses of antibiotics. By the time they're 10, they've received about 10 courses of antibiotics. During pregnancy, just before the intergenerational transfer of the microbiota, in the US, more than 50% of women are treated with antibiotics or given antibiotic prophylaxis. There is also antibiotic exposure from the use on the farm, and we don't really have any idea what the magnitude of that is, but it's been going on for 70 years. Antibiotics in the United States require a prescription. In many parts of the world, no prescription is required. This is from a small village in the Peruvian Amazon where the antibiotics are available generically over the counter, all manufactured in China, I might add, and readily available to anyone who's willing to pay the pharmacist. Now, recently, the Gates Foundation has sponsored a study of 8 birth cohorts in developing countries, the MAL-ED birth cohorts. And this slide looks at antibiotic use in the first 2 years of life. So on the x-axis is age in childhood. That's the first 24 months of life. The y-axis is antibiotic courses per person year. Here are 8 communities. And I just happened to draw in the line for the US. This is what it would look like for US children. And in 6 of the 8 communities, the antibiotic use is higher in these developing countries, or a lot higher. And in fact, in these communities in Bangladesh and Pakistan, the use averages out to about 10 courses per year for these children. This is extraordinary, the amount of antibiotic use. People in many parts of the world consider antibiotics miraculous and life-saving. So when their child is ill, they give their child an antibiotic. So the magnitude of the exposure in developing countries is really quite extraordinary. And this of a number of scary slides, this is one of the most scary. So what I want to talk about for the major part of my talk are what I would call paradoxical effects of antibiotics. I'll show you evidence that antibiotics can affect metabolic and immunological development. We have evidence that antibiotics can increase susceptibility to subsequent infections, but this hasn't been published yet, so I'm not going to show it today. We'll show evidence that the effects of antibiotics can be passed on to the next generation via the altered microbiome. And I'll give an example of how an altered microbiota in young children is associated with increased risk of subsequent disease. So first I want to talk about antibiotic use on the farm, because farmers about 70 years ago discovered that if they gave low doses of antibiotics to their farm animals, it would promote their growth. It worked from chickens to cows, a wide swath of vertebrate evolution. The reason that farmers are giving the antibiotics is because it works to promote their growth. It's been found to work for just about any antibacterial, regardless of its chemical structure, class, target, or spectrum. Antivirals do not work. Antifungals do not work. Importantly, the earlier in life the antibiotic is given, the bigger the effect on growth rate, the bigger effect on feed efficiency, the conversion of food calories into body mass. This is what farmers are trying to do. So based on these observations, it suggested to us that antibiotics were perturbing a developmental phenomenon. So we began to do studies in mice in which we would give mice antibiotics or not, measure the properties, analyze the microbiome, and look for a relationship. So now, in most of the rest of the talk, I'm going to show you the results of these mouse experiments. The first studies were done by Il-sung Cho when he was a postdoctoral fellow in my lab. Il-sung gave mice 4 different antibiotic regimens at the midpoint of the FDA-approved dose on the farm. And here we're measuring percent body fat. Here's the control group, no antibiotics. The mice that got the antibiotics put on more fat. This was our first evidence that antibiotics was changing metabolism in mice. We had lots of other evidence in the liver and other organs and in adipose tissue. But I'm going to go on to the next study, which was led by Laurie Cox when she was a graduate student in the lab. Laurie asked, what are the effects of combining a high-fat, high-calorie diet and antibiotics? So she gave mice the low-dose antibiotic exposure used on the farm. In this case, it was penicillin, or not. All the mice were started on normal chow, and at week 17 of life, half the mice were put on a high-fat diet. We studied male mice and female mice, total mass, fat mass, and lean mass. Starting with the male mice in the group and total mass in the group that received no antibiotics, normal chow, that's this black line. Normal chow with antibiotics, they're bigger, just like on the farm. High-fat diet, bigger still. High-fat diet plus antibiotics, big est. Lean mass increased on antibiotics, like on the farm. Fat mass increased on the high-fat diet, even more on high-fat plus antibiotics. Female mice, many of the same trends. Let me point out that for fat mass, the average female mouse on the high-fat diet gained 5 grams of body fat, high-fat plus antibiotics, 10 grams. The addition of the antibiotics doubled the amount of fat accumulated. The antibiotic potentiated the effect of the high-fat diet. We saw similar changes in the liver. So up to this point, the mice were getting antibiotics for their whole life. So Laurie then did an experiment to see, are the changes durable with limited antibiotic exposure? So in addition to no antibiotics and lifelong antibiotics, mice got only 8 weeks or only 4 weeks of antibiotics. Here are the results on total, lean, and fat mass. The black line is the control group. What Laurie found is that all 3 groups of mice exposed to antibiotics had increases in total, lean, and fat mass. So 4 weeks, starting early in life, was sufficient for the full effect. We were interested in what was going on in the intestine. And here's work by Jackie Leung in Peng Lok's lab looking at the effects of the stat antibiotics on intestinal Th17 populations. Here we're looking at the small intestine, the large intestine by flow cytometry. No matter how we measure it, Th17 is down. And we've found this quite consistently across many studies, which I'll show you subsequently. So there's an effect on the T cell populations in the intestine. Now, what about the microbiota? Here we're going to look at the fecal community structure of mice at 3 weeks of age. And at 3 weeks in this experiment, there were control mice not receiving antibiotics, 3 groups of mice receiving antibiotics, and at 3 weeks, all 3 of them were receiving the antibiotic. In this principal coordinates analysis, black circles are control, orange are receiving antibiotics. There's a lot of diversity. They overlap a lot, but they're a little different, which is not surprising. One group is getting antibiotics, the other group is not. Now we're going to turn to 8 weeks. And this is maybe the second scary slide. So at 8 weeks, we have 3 groups of mice. No antibiotics, continued antibiotics, or 4 weeks of antibiotics, and then we stop the antibiotics. So now we look at the community structure. Here are the controls in black, continued antibiotics in orange. They're more separate than before, which is not surprising. One group is continuing to get antibiotics. But the group that stopped antibiotics, their microbiota have reverted to normal. So the effect of the antibiotic was transient on the mice, on the microbiota, but the effect on the phenotype was permanent. So this provided evidence that altering the microbiota at a critical point in early life had long-term consequences. We continued this work by seeing whether we could transfer the phenotype with microbiota alone. So from mice that received antibiotics or not, we harvested their cecal contents and we gave these to germ-free mice, and we followed them for 5 weeks. These mice did not receive any antibiotics. Here we're looking at total, lean, and fat mass. The black line is the control group. we found that the antibiotic-perturbed microbiota caused the mice to have an increase in total mass, no change in lean, and an increase in fat mass. So we have evidence here that the signal is in the microbiota. This is not an antibiotic effect, it's a microbiota effect. We wanted to see, were there immunological effects as well? So here we're looking at expression of genes involved in intestinal defenses. Here are the donor mice that had received antibiotics or not. We're looking at 3 genes related to Th17, transcription factors, cytokines, 2 antimicrobial peptides downstream of Th17. Here we see the same kind of depression that we had seen earlier, very consistent findings. So now the question is, what about the mice, the germ-free mice that received the microbiota? And the results are shown here. We see the same kind of trend. So we have evidence from this study that the immunological phenotype is transferable by the microbiota, by the altered microbiota as well. Now, up to this point, the model is the STAT model, low doses of antibiotics every day in the drinking water of the mice. But Yael Noble, a student in the lab, developed a second model, which we called PAT, for pulsed antibiotic treatment. This was based on the kinds of courses that human children would get to treat their ear infections or their throat infections, therapeutic doses in 3 short pulses. And her hypothesis is that this would affect phenotype. I don't have time to show you her results, but I wanted to introduce the model and credit Yael for that. I want to turn now to the study by Victoria Ruiz, who followed up on Yael's study. And in addition to a model with 3 pulses, she asked what happens if she just gives the mice a single course of antibiotics early in life. Well, here we're looking at a PCOA, again, of the microbiota. This is at day 52 in life. This is 40 days after the single antibiotic course ended. Here's the control group. Here's 3 courses of antibiotics. Here's 1 course of antibiotics. With a therapeutic course of antibiotics, the microbiota do not normalize. It's different than the subtherapeutic. So there's a much more prolonged effect from antibiotics given to mice early in life. What's the effect on the host? Here we're going to look at the effect on ileal gene expression and immunological effectors. This is a heat map of a nanostring encounter assay of 547 genes involved in inflammation and immunity. It's an unsupervised hierarchical clustering. This is the group of mice that didn't receive antibiotics. They cluster by themselves. The 2 groups of mice that received antibiotics, 1 or 3 courses, they formed the second cluster. You can see that there's a lot of similarity. In total, 147, about a quarter of the genes are differentially expressed between antibiotic and control, mostly with 3 courses of antibiotics, but about half of them with only a single course of antibiotics, involving important genes like the polymeric immunoglobulin gene receptor, down with 3 courses, down with 1 course. Intestinal secretory IgA goes up in normal mice, but doesn't go up as rapidly in mice given 1 course of antibiotics, and even worse with 3 courses. Th17 down with even a single course of antibiotics. So in this experiment, we were able to, the mice received the mom mice received the antibiotics in their drinking water. So we were able to ask what happens, how does the antibiotic pulse affect ileal gene expression in the mother and her pups? They got the antibiotics at the same time, and now we're studying them 40 days later. And this is a volcano plot from RNA-seq of ileal gene expression in the PAT or control dams or the pups. In the dams, 40 days after the antibiotics, there are 106 genes that are differentially expressed. But in the pups, there are 1,300 genes that are differentially expressed. So antibiotic exposure at the same time point and assessment at the same interval shows that there's much more profound effect of the same antibiotic in the pups than in the dams. Now, is that an antibiotic effect or a microbiome effect? And is the microbiome necessary for the effect? So to answer that question, we gave the antibiotic to germ-free mice or not to see if there would be effects on gene expression. And as a control, we gave the antibiotic or not to conventional mice and studied them 40 days later. Here's the nanostring heat map again. Here are the normal mice, no antibiotics. They cluster all by themselves. Here are the germ-free mice, whether they get antibiotics or not, they cluster together. Here are the conventional mice with antibiotics. They're actually clustering with the germ-free. You can see it a little better here on this coordinate map. Here are the control mice, no antibiotics. Here are the germ-free mice, whether they had antibiotics or not. Conventional mice with antibiotics. Effect on Th17 in the conventional mice, nothing in the germ-free mice. So this provides evidence that having a microbiota is necessary for the effect. The effect is going through the microbiota. So the question is the antibiotic-altered microbiota sufficient to affect immunity? Well, Isaac Engstrand studied that when he was visiting the lab. Mice were given a macrolide for 5 days. 2 days later, they were sacrificed, their ileal contents harvested. The mice were and given to germ-free mice again, and now we followed these mice for the next 11 weeks. And we asked what happens to their immune function. And here are two markers. Here's the proportion of splenocytes that have the TCR beta marker. It's down and pat 11 weeks after the transfer. Here's fecal IgA, which goes up normally in the mice that got the normal microbiota, reaching a high plateau. It goes up more slowly, lower plateau. So we have evidence for this, again, that the signal's in the microbiota, and that's sufficient to have immunological changes. Now those are based on markers. We also did a study of disease, and this is work that Alexandra Levanos did when she was an MD PhD student in the lab. Ali's hypothesis is that antibiotics would promote type 1 diabetes in NOD mice. These are mice that spontaneously get type 1 diabetes through effects on gut microbiome, changes in intestinal gene expression and T-cell subtypes leading to type 1 diabetes. Ali actually showed every part of this hypothesis, and now we're continuing to work on this as a model of antibiotic-perturbed type 1 diabetes. Now, I next want to turn to inflammatory bowel disease, because there are studies that show that antibiotic exposure is associated with increased risk of developing IBD. This is from a large epidemiologic study in Denmark involving all the children born in Denmark over a 9-year period. And what the investigators showed is that the more courses of antibiotics that the child received, the more likely they were to develop IBD, either Crohn's disease or ulcerative colitis. So we wanted to know, could an antibiotic-altered microbiota affect IBD outcome? And furthermore, we wanted to see if there could be an effect in the next generation. So to examine that, we studied germ-free C57 mice that were either wild type or IL-10 deficient. And IL-10 deficient mice spontaneously develop colitis. And so this work was done by Angel Schulfer, who was a graduate student in the lab. She studied germ-free wild type or IL-10 deficient mice, and she either gave them an antibiotic-perturbed microbiota or a control microbiota. As I said, these mice were pregnant. They were conventionalized, so there were two genotypes, 2 inocula. We had 4 groups of mice. The pregnant mice soon gave birth to pups, and now we followed the pups till they were middle-aged, up to age 21 weeks in life. And basically, we asked 2 questions. Does the perturbed microbiota have an effect on the ecology of the microbiota in the pups after going through their moms? And is there an effect on the subsequent disease? We showed that there were many effects on the ecology. I'm just going to show you one effect. Here we're looking at the stability of the microbial community in the recipient mice, whether they got the control inoculum or the antibiotic-perturbed inoculum, whether they were wild type or IL-10 deficient. And here we're looking at the Jacquard analysis of distances. The higher the distance, the greater the instability of the community. And in each case, the antibiotic-perturbed microbiota led to increased instability, first in the mothers, then in the pups. This is one of a number of ecological changes that we found. In general, there was transfer with high fidelity from inoculum to mother to pup, but the fidelity was less with the antibiotic perturbation. But the really important question is what happened to disease in the IL-10 deficient mice? And that's represented in this slide. Here we're looking at colonic pathology in the IL-10 deficient pups at week 21 according to the microbiota to which their mothers were exposed. Here's an example of a pup whose mother got the control microbiota, and here's an example of the mother who got the antibiotic-perturbed microbiota. You can see there's a huge difference. And in fact, when we measured it, the distance was 55-fold. It's a huge phenotype. So to summarize this experiment, what's important to remember is that these pups never saw antibiotics. Their mothers never saw antibiotics. All they saw was a perturbed microbiota. So that means that the enhanced disease signal is entirely microbial. It also provides evidence that the antibiotic effect can cross generations, can go into the next generation, and also introduces the concept that inheritance may also be based on microbes and their genes, not just host genes. Now, earlier, Maria Gloria showed the work that we had done following a birth cohort in New York, and this is work that Nick Bakulich performed. And what we found over the first 2 years of life is a very orderly succession of the microbiota, with certain organisms coming in first, the Enterobacteriaceae, Bifidobacteria, Bacteroides, etc. So, and this is consistent with a wide body of work of this orderly succession. So we wanted to look at a disease situation to see what this might look at. And so we were able to work with a group in Denmark, the investigators of the COPSAC study. This is the Copenhagen cohort of asthmatic disease, working with Jacob Stockholm, who did a postdoc in my lab as a visitor. So here we're going to look at the microbial compositions in feces in a cohort of more than 600 children in Denmark. And the children's microbiota were sampled at 1 week of age, 1 month of age, or 1 year of age. And here we're looking at the beta diversity by sampling time. The 1 week and the 1 month are pretty similar. The 1 year is quite different. So there's been a maturation of the microbiota. When we do PAM clustering, we can see 2 distinct PAM groups, which we call PAM1. This is the, well, it's PAM1 and 2. And now when we compare sampling time and PAM cluster, we see that at 1 week, most of the kids' microbiome is in PAM cluster 1, a few in 2. By 1 month, a few more. But by 1 year, the microbiota has largely shifted to this second cluster. And so now we're going to ask, what's the relationship of the beta diversity of the microbiota in the samples from 1-year-old kids in relationship to whether the child developed asthma by the age of 5? So when we look at all the kids in the study, in this case nearly 600, there is a significant difference in the 1-year microbiota between the kids who later developed asthma or not. If we now substratify and say, what in the kids who are born to non-asthmatic mothers, is there an effect? And the answer is no. But in the kids born to asthmatic mothers, we can see a very significant and dramatic effect. So the combination of being born from an asthmatic mother and the composition of the microbiota appears to be related to the risk of developing asthma in the child. We can look at this using a Kaplan-Meier estimate of cumulative risk. So across all the children in the study, kids whose microbiota at 1 year were in PAM cluster 1, that is dismature, they had an increased risk of asthma. Their odds ratio was almost 3. When we looked at the kids born to non-asthmatic mothers, there's no relationship at all. But in the kids born to asthmatic mothers whose microbiome was in PAM cluster 1 at the age of 1 year, the odds ratio for developing asthma was 13. A very, very high rate. This is similar to the rate of the odds ratio, let's say, for smoking and lung cancer. It's a very significant association. Now, one might say PAM clusters are somewhat arbitrary, so we also looked at microbiota by age scores, or MAZ scores. We divided the kids into whether they had the most mature, the top half of maturity, or the bottom half of maturity. We see in the less mature, higher rates of asthma, odds ratio about 2, no effect in non-asthmatic mothers, full effect in the asthmatic mothers, odds ratio of about 6. So we're interested in what are the microbes that might be related to this risk of asthma, the microbes present at 1 year that might be related to the risk of asthma at 5 years. we don't see much of significance in the kids born of non-asthmatic mothers. We see a number of significant findings in the kids born of asthmatic mothers. This is a little too small and too busy for you to read. But I want to point out that Brett and his colleagues had done a study, a very similar study in British Columbia, and they identified 4 taxa that were associated with protection against the development of asthma in British Columbia. We detected 3 out of the 4 from there. So there's high concordance between these 2 studies, suggesting that the nature of the microbiota is a predictor of which children are going to get asthma in this group whose mothers are asthmatic. So let me just summarize to bring this talk to an end. Our idea is that there are cumulative effects on the early life microbiome. There are effects which I call deep inheritance in middle life history. This reflects those prior generations of mothers, grandmothers, and great-grandmothers, and the prior antibiotics and antibacterial exposures. Then there's a period of pregnancy and perinatal time when there's antibiotics, C-section, and washing are differentially selecting for microbes. Then there's the postnatal early childhood period where kids are getting antibiotics, their mothers are getting antibiotics, and they're reaching them via breast milk. Kids are getting formula. Kids are exposed to antibacterials all the time. And our idea is that this is changing the ratio between the ancestral microbes and, let's say, more modern acquired opportunistic commensals. And why is that important? It's because in the gut, there's an interface where the microbiota in the lumen is talking to host tissues. And this affects events in the portal circulation involving the liver and adipose tissue, involving lymphoid populations in the lamina propria, involving the vagus nerve, hormones and neurotransmitters, involving brain development. So that's the important interface. But however we look at it, the hypothesis is that we are losing diversity. This is a schematic that we made a couple years ago showing that, in fact, in a developed country like the United States, we presume that we've been losing diversity all along. Here's 1950. This is basically when the antibiotic era began. But there have been changes in our ecology before that, like chlorinated water and other factors. This might be a country like India or China that started develop modernization later, but is catching up in a hurry. And this might be a country in Latin America or Africa just beginning to modernize. So this was kind of a view of diversity in 2016. And the really important question is what's going to happen from now? Is our microbiota going to further decline in diversity? Are we going to be able to develop ways to arrest the decline? Or will we reverse through restorative steps? And like Maria Gloria, I think that the only way that we will combat the loss is through restoration. And what I would propose is that the doctors of the future are going to conduct new analyses of child health. When they're examining the baby, they will also examine the baby's diaper. And they will analyze, does this baby have all the global microbes that a healthy baby should have? Do they have the personal microbes that a baby of that genotype and other phenotype markers have? And if these babies do not have such microbes, then the doctors of the future are going to administer those organisms that we think are beneficial and have been lost, and ultimately observe whether there is improvement of phenotype, an optimization of health, and along the way, perhaps do other steps to reshape the microbiome to optimize health. I've recognized many of the people who've done the work that I've shown today, and I in particular want to recognize those people in bold whose work I particularly showed. Lots of other people helped in this work, and support by NIH and other agencies. And thank you all very much for your attention.